Hello. So we are going to talk about salivary metabolome, which we already saw in the seminar about the salivary metabolic hormones. So this article with the title "A Repository of Salivary Metabolome and its Key Drivers in 1,436 European Children" was published in the journal Biomedicine, which is a part of the Lancet Discovery Science, with an impact factor of 10.8 in the year 2025. So the novelty of this study. So the oral cavity is host to both microbial and viral cells. The salivary metabolome comprises exogenous and endogenous metabolic products, the latter by inflecting the circulating metabolite levels via passive diffusion from blood. So saliva thus presents superior properties to blood as it provides a mirror to the interactions between microbiome, diet and the host metabolome. So no studies report on salivary metabolome and its key drivers in relation to health. And the first study to comprehensively characterize the salivary metabolome of children and adolescents, analyzing samples from 1,436 participants across 4 independent European pediatric cohorts, which is from Belgium and Finland. So the aim of this study was to study the extent to which the salivary metabolome reflects age and anthropometric parameters, particularly weight status, as well as indicators of mental well-being and lifestyle, and how food composition and microbial composition contribute to that. Here the methodology was, they used pediatric cohorts which into two types, that is exploratory cohorts and independent validation. So already there were cohorts of study which were done in the Finland and Belgium using the European kits, which was the FAME, which is conducted from 22 and 23, the FINFIT from 2011 and 14, the PANIC, which was conducted from 2007 to 2009, and OPERA from in March 2018 to August 2018. So the 4 cohorts were studied. They collected the saliva, they did the metabolic profiling and the fingerprinting, and they found out how the metabolic profiling, the results were correlated with the other parameters which were examined in this study. So the anthropometric measurements, including the height, weight, waist circumference and body fat percentage, were taken using standardized methods by a trained professional, and the BMI was calculated and adjusted for age and sex according to the Belgium and Finnish reference standards. Then they collected data about the sleep duration, the physical activity, total screen time, food consumption, depressive symptoms and self-esteem. So the included cohorts represent both population-based and more dedicated sampling strategies, enabling the investigation of salivary metabolic phenotype associations across a broad spectrum of weight statuses. So they did group comparisons, stress score construction and correlation analysis. The salivary sample collection was from all the 4 cohorts. They did a passive drool collection, unstimulated saliva in 3, and the other one they collected under supervision of trained staff using the passive drool collection. One was through stimulated saliva collection, which were the patients the participants stood paraffin to stimulate the saliva and collected the samples. Before the sample collections, participants were required to refrain from eating or drinking for at least 3 hours before collection and could only brush their teeth in the morning. The samples were immediately stored at -12 to -20 for up to 1 week. The salivary metabolome targeted profiling and untargeted fingerprinting was done using the HRMS full scan. The V3-V4-16S RNA sequencing for salivary microbiome analysis were done. The statistical R package was used for cross-cohort consistency, mental well-being and diet harmonization, and interpretation of correlations and significance. So all these things were done and the correlation was found. So the results showed that the pediatric cohort characteristics was across all the 4 cohorts, found that the girls showed a poor mental well-being and high body fat percentage in all the 4 cohorts. And the correlation between children with adolescent salivary metabolome and their age, anthropometrics, mental well-being, lifestyle. In the exploratory cohorts, they found the correlation for the salivary metabolome and the mental health. So salivary metabolome variation was most strongly linked to anthropometrics and age, with functional pathways involving amino acids, purine, pyrimidine, glutanoate, nitrogen and vitamin metabolism, and small association observed with depressive feelings. In FIT cohort, they found the metabolome correlation were dominated by age, BMI, weight to height, and the pathway enrichment highlighting the amino acids alongside strong body image related metabolic signatures, modest association with the physical activity, and weekday sleep duration. And the metabolic indicators for overweight and obesity detected in saliva across all cohorts. They found all the 4 cohorts the different kinds of metabolites which were found, and many were novel in this, which was related to the anthropometric measurements. And salivary metabolites related to mental well-being and lifestyle across all cohorts show that in fame, the depressive feelings, perceived stress, and external eating correlated with 35 metabolites, with 4 showing significant differences across psychological stress levels. In the film, it 11 metabolites including L-methionine, L-tryptophan, all those things were related with the body image, self-esteem, and sleep duration. So all the 4 cohorts showed the difference in the metabolic profiles and their correlation with the mental well-being. The screen time showed no metabolic association in FinHit, while the physical activity correlated positively with the L-glutamic acid and negatively with N-acetyl use. The latter displaying an opposite correlation with the panic. And artificial sweeteners consistently detected in pediatric saliva and their association with food intake, weight status, and mental well-being. So the artificial food sweetener was mostly in the dietary drinks and savory snacks. So all these things correlated in all 4 cohorts. So in conclusion, the study shows that the age and weight status are the strongest determinants of salivary metabolome, with several metabolites repeatedly linked to body weight and mental well-being across multiple cohorts. And many of these associations align with previously reported blood-based metabolite findings, reinforcing the reliability of these tests. At the same time, most of the identified salivary metabolite relationships are novel and offering valuable insights into future biological studies and providing a foundation for designing interventions that target salivary metabolic pathways in health and disease. So the novel salivary metabolite they found were the acesulfame K, which linked to diet carbonated drinks and soft drinks, and L-aspartyl, which is also due to this carbonated drinks. Indole acetic acid, the decreased levels associated with high chocolate-based food intake, and thymine lower levels with high chocolate base. So the dietary drinks chocolate-based, and the hypoxanthine, theobromine, and caffeine, all they were related to the novel metabolome which was found. So saliva offers a powerful non-invasive window into metabolic health, integrating hormones and metabolites as emerging biomarkers. The limitations of the study was a cross-sectional limitation was there. Generalizability issue because the non-European or diverse population was not included, and mechanistic uncertainty, pathway links and speculated here. In the future scope, longitudinal studies, intervention studies, and mechanistic research. And this was the observational study. So this stroke guidelines were followed in this study, which was with a score of 7.2 out of 10, which had a good descriptive quality, moderate methodological robustness, which was strong in rational methods and results, weaker in bias control, sample size, reasoning, and confounder handling. Thank you.